Neurotransmitters. The chemicals neurons use to communicate between them when you run, sing, or talk about this guy.
Money, I want more money.
And what a better way to present some of them, an iceberg video. As in every iceberg video format, I will cover at the beginning the more famous neurotransmitters, and further down we go, the more obscure the neurotransmitters become. At the top of the iceberg lies dopamine. Chances are you reach this video simply because you watched some dopamine detox video. Dopamine is synthesized from phenylalanine, which is oxidized to tyrosine, which is oxidized to dopa, which gets rid of its carboxyl group to become dopamine. Then it is incorporated into synaptic vesicles via a vesicular monoamine transporter. The synaptic vesicles fuse with the membrane to release its precious cargo, which activates G-protein coupled receptors, which trigger different changes in different neurons. To avoid a constant exposure of receptors to dopamine, the latter one is removed from the synaptic cleft by a natrium-dependent cotransporter on surrounding glial cells and on nerve terminals. Both neurons and glial cells contain two enzymes which degrade dopamine: monoamine oxidase, shortened MAO, and catechol O-methyltransferase, shortened COMT. Some of you might want to bring MAO and COMT to make order in your brain, but there is a bigger picture. In patients with Parkinson's disease, dopaminergic neurons are lost. Thus, we infer dopamine has a role in motility, and the dopamine detox phrase should not be adopted word by word. Epinephrine. Why did you include this almost unknown so high? The hormone of fight or flight response. Wait a second, why is it into a neurotransmitter iceberg video? It is because, besides acting as a hormone, adrenaline also acts as a neurotransmitter, albeit not in the higher regions of the brain as dopamine does. It follows a similar cycle as dopamine: integration into synaptic vesicles, release into synaptic clefts, activation of G-protein coupled receptors, removal from synaptic cleft. But epinephrine neurons are mostly involved in regulating respiration and the heart. Serotonin. The happiness molecule, as many would say, although low levels of serotonin alone cannot induce depression, what fewer people know is that serotonin is important for circadian rhythm too, because it can be used to produce melatonin, the hormone that puts you to bed. Hopefully not during my video. But what is a precursor of serotonin? It turns out it is tryptophan, one of the essential amino acids. The rest of the cycle is similar to other neurotransmitters: incorporation into vesicles, release into synaptic clefts, activation of G-protein coupled receptors, removal from synaptic cleft, and degradation by MAO. Acetylcholine. This is one of the best studied neurotransmitters. One can find it in both central nervous system and at the neuromuscular junctions. It is synthesized, as the name suggests, from acetyl-CoA and choline in a reaction catalyzed by the choline acetyltransferase. Unlike the biogenic amines we covered before, acetylcholine is used differently. Its uptake into synaptic vesicles is regulated by a protein that exchanges acetylcholine for protons because the interior of the synaptic vesicles is highly acidic. Moreover, there is no reuptake of acetylcholine as it is broken down into acetate and choline directly in the synaptic cleft. Nevertheless, choline is incorporated back into neurons via a natrium-plus-dependent cotransport. Because it is so widespread, there is more diversity in the acetylcholine receptors. Some named nicotinic receptors, because nicotine can also be them, are channels that open in the presence of acetylcholine, enabling different cations to enter the postsynaptic neuron. Others, named muscarinic receptors are G-protein coupled receptors that activate or inhibit potassium channels. Histamine. I have seen it before, but I don't remember where. Maybe here. It is synthesized from an essential amino acid called histidine in a reaction catalyzed by histidine decarboxylase. Similarly to other biogenic amines, it is incorporated into synaptic vesicles, released into synaptic clefts, activates G-protein coupled receptors, and is degraded by histamine methyltransferase and MAO. However, different types of receptors can influence different functions, like equilibrium in the case of H1 receptors or gastric secretion in that of H2 receptors. Glycine. The smallest amino acid. It is synthesized from serine, which at its turn is synthesized from 3-phosphoglycerate, a glycolysis intermediate. Then it is packed into synaptic vesicles via a vesicular inhibitory amino acid transporter and released into the synaptic cleft. It binds to and opens channels that let chloride and other anions enter the postsynaptic neuron, leading to hyperpolarization and thus inhibition of the postsynaptic neuron. An antagonist of glycine, strychnine, maintains the channel closed. This is how many of inhibitory synapses in the spinal cord work. Glutamate. Another amino acid, but in contrast with glycine, it is a notoriously excitatory amino acid found in the central nervous system. It is synthesized from glutamine by mitochondrial glutaminase, as glutamate cannot pass through the blood-brain barrier. It is incorporated into synaptic vesicles by vesicular glutamate transporters, then released into the synaptic cleft. It is removed from the synaptic cells by Na+-dependent cotransporters into the glial cells, which convert glutamate to glutamine, which is then incorporated into neurons. Most of the types of glutamate receptors are channels that allow passage of cations, especially sodium and potassium. Some of them, the NMDA receptors, have a more complex mechanism of opening, which is also regulated by the membrane potential. At highly negative membrane potentials, a magnesium ion blocks the channel, but when the membrane is depolarized, the magnesium ion is expelled and the channel is open. In many cases of schizophrenia, this receptor functions below its normal capabilities. There are also some metabotropic receptors that can produce inhibition of postsynaptic neurons. GABA. Despite the fact that it is synthesized from glutamate, GABA or gamma-aminobutyric acid is mostly an inhibitory neurotransmitter. It is transported into vesicles via the same transporter as glycine. It is released into the synaptic cleft, from where it is removed by a Na+-dependent cotransporter, such that it can be degraded into succinate by GABA transaminase and succinic semialdehyde dehydrogenase. GABAergic neurons can be found in cerebellum, such as these skull-looking Purkinje cells. Norepinephrine. This structure looks a bit familiar now, unless you skip the top of the iceberg. Yes, it resembles with dopamine, because the latter one is used to synthesize noradrenaline, and yes, it resembles with adrenaline, as adrenaline is made from it. Norepinephrine is another hormone that can also act as a neurotransmitter, especially in this area of the brain, having a role in circadian rhythm, attention, and coordination of smooth muscles. It has the same cycle as the other two catecholamines. I won't bother repeating it. Endocannabinoids are more similar to lipids than to other neurotransmitters. Some of them include anandamide and 2-arachidonylglycerol, which are produced from the degradation of lipid membranes when calcium concentration increases in the postsynaptic cells, which happens when the cell is depolarized. Then endocannabinoids are released from the postsynaptic neurons to bind the CB1 receptors on the presynaptic neurons. These receptors are coupled with G protein such that the release of GABA from the presynaptic neuron is inhibited. This is a really smart tactic for active neurons to prevent their inhibition by other neurons. Nitric oxide. It is more likely that you heard about it in a chemistry class or in a geography one rather than a biology class, which is one of the reasons why it is so close to the bottom despite many people searching about it. Another reason is that it can also be viewed as a secondary messenger whose synthesis from arginine is catalyzed by an enzyme called NO synthase whose regulation depends on cytoplasmic calcium concentration. However, NO is considered a neurotransmitter because of its small size. It can cross the plasma membrane and it can propagate its effects beyond the cell where it was produced. Finally, at the bottom of the iceberg, something 99% of you wouldn't search on YouTube, an 11 amino acid peptide called substance P. It has an important role in transmitting information about pain and temperature. Its release is inhibited by opioids, which is the reason why they can suppress pain. And these are some of the most common neurotransmitters. Write in the comments about other neurotransmitters you know, and if you enjoyed the video, don't hesitate to hit the like button and subscribe to more neuroscience content. My name is Mihai and you watched Studying at Uni.